DNA, first of all, the helical model of the DNA first observed and by the Watson and Crick in 1953. Therefore, this structure of DNA is also called as Watson and Crick model of DNA. Now, Watson and Crick model of DNA in eukaryotes, the Watson and Crick model, that is Watson and Crick observed how is the structure. They observed that the DNA is the spiral ladder-like structure having two strands which are running opposite in direction but parallel to each other, so called as anti-parallel strands. Now, how is the structure? Try to understand. One of the imaginary axis is there. To this imaginary axis, there are two strands. One strand is in upward direction, that is ascending, and other strand is descending strand. In Watson and Crick model, the DNA strands are running parallel to each other. This is the one of the ascending strand, this is the descending strand. Okay. Now, these two strands are going to parallel to each other but twisting at a imaginary some axis, central axis. They are twisting to each other and hence looks like the spiral ladder-like structure. You know, ladder means the steps we want to go upward. That like structure here seen by the Crick and Watson. Now, these two strands are linked with each other with the help of, or they get bind with each other with the help of weak hydrogen double and triple bonds. Now, these double and triple bonds are just connecting or winding the two strands or binding the two strands to each other. Now, this spiral ladder-like structure followed by make up the strand. Each strand now made up of three chemical components. That is, DNA is made up of three chemical components. That is, first of all, the pentose sugar, then nitrogenous bases, and phosphate. Now, pentose sugar. In DNA, the pentose sugar is of deoxyribose type. Deoxyribose type of pentose sugar is there. Now, each strand of the DNA is made up of many nucleotides or monomers linking to each other. Now, so that DNA molecule is polymer structure. Many monomer or many nucleotides are linked together with the help of bond, that is phosphodiester bond, linking to each other of the nucleotides and forming the polymer structure. Now, each strand, how the link or how it is made up, that is we want to study here. First of all, what happened? There is a pentose sugar. Suppose this is pentose sugar. If we giving the number out to the pentose sugar, the clockwise direction, the carbon number 1, carbon number 2, carbon number 3, carbon number 4 and fifth carbon is out of the ring. Now such 5 carbons are there in the pentose sugar, so it is called as pentose sugar. Now to this pentose sugar, to the first carbon, when there is attachment of any nitrogenous base, any nitrogen base, as we know that the nitrogen bases are of two types: purines nitrogenous bases and pyrimidines nitrogenous bases. Purine nitrogenous bases are double ring structures, whereas pyrimidine are single ring structure nitrogenous bases. Now purine nitrogenous bases are adenine, that is A, and guanine. In DNA, purine nitrogenous bases are adenine and guanine, whereas pyrimidine nitrogenous bases are thymine and cytosine. Now, these 4 nitrogenous bases, that is 2 of the purines and 2 of the pyrimidines, out of that, out of the 4 nitrogenous bases, any one nitrogen base is attached to the first carbon of the pentose sugar, then this structure is called as nucleoside. The attachment, that is bond between first carbon and nitrogenous base, is called as glycosidic bond. The bond between first carbon and sugar molecule is glycosidic bond. Now, to this nucleoside, whenever there is attachment of phosphate group from the phosphoric acid, that is at the 5th carbon, when P is attached to the nucleoside, the structure is now called as nucleotide. Are you what? When to the nucleoside means pentose sugar and nitrogenous base, this total structure is nucleoside. To this nucleoside, whenever there is attachment of phosphate group at the 5th carbon, then the structure is called as nucleotide. Now this nucleotide structure is also called as monomer. The bond between the sugar molecule and phosphate group, that is this bond, the linkage of the sugar to the phosphate, this is called as sugar phosphate bond. Sugar phosphate bond. Okay. Now this sugar phosphate bond linking the P group to the pentose sugar and glycosidic bond linking to the nitrogen base to the first carbon of the same sugar. Now whole structure, the pentose sugar, nitrogen base and phosphate group get together called as nucleotide. Now this each nucleotide is also called as monomer, so that a single strand of the DNA is made up of such a many nucleotides linking together. So the DNA is called as polymer structure. Many monomers linking, so called as polymer structure. Now molecular structure we want to see of this DNA, of the same DNA. Diagrammatically, this how the two strands are linking. See, two strands parallel, but they are opposite in direction, so that the pentose sugar here also we are drawing here in opposite direction. Now nitrogen's base, suppose this is adenine, here with thymine. Suppose this is a guanine, here with cytosine. Suppose this is repeated thymine, again pairs with the adenine of this one, then cytosine here with guanine. To this 5th carbon, as we know that there is attachment of phosphate group. Here also phosphate group is to be attached to the fifth carbon. This is the ascending strand, that is this one, and this is the descending strand. Now, this figure is not yet completed. The nucleotides are just bind to opposite complementary nucleotides, but the same strands nucleotides are not linked together. Now, this nucleotide get linked with the adjacent. This also get linked with the adjacent. This also get linked with the adjacent. Likewise, here also. Means one nucleotide get linked with of the same strand linked with another nucleotide with the help of a bond between carbon number 3 and successive phosphate group, and this is called as phosphodiester linkage. Phosphodiester linkage. Now, with the help of phosphodiester bond, the two adjacent nucleotides are get linked in all over into the each strand, so that each strand is get turns into a polymer structure by linking. The link between these two is nothing but the phosphodiester linkage. Whereas this is the one strand and this is the another opposite strand which is running parallel to each other. Now these two nucleotides are get attached, these two strands are attached to each other with the help of double or triple big hydrogen bonds. Whenever in DNA, always one purine base always pair with one pyrimidine base. That is, adenine is purine base, thymine is pyrimidine base. Guanine is purine base, cytosine is pyrimidine base. It means that there is a ratio of purine and pyrimidine bases is 1:1, maintained here 1:1 in the DNA. Because of this nature of these fixed or specific nitrogenous base pairing, adenine always pair with thymine, or thymine always pair with adenine, or guanine always pair with a cytosine, and cytosine always pair with a guanine. This nature of the DNA strand indicates that if we have the sequence of one strand of the nitrogenous bases, we can imagine or we can noting that the another strand sequence also. It means that the nitrogen base pairing is complementary to each other. The bases are complementary to each other. Now, we want to come back to this eukaryotic DNA molecule. See, this is the spiral ladder like structure, no doubt. To the imaginary central axis, the DNA strands are going to twist into each other. Looks at spiral ladder like structure. DNA strands are the base pair. We already studied that DNA strand base pairing is specific, that is one purine base is always pair with the another pyrimidine base. They are complementary to each other. Then in DNA, the length of one spiral is 34 angstrom. In one spiral there are 10 nitrogenous bases are present at equidistance, that is 3.4 angstrom in it. The diameter of the DNA strand is 20 angstrom. The 10 nitrogenous bases are of the two strands are placed at equidistance, that is 3.4 angstrom in it, and these 10 nitrogen bases including the two loops of the DNA, that is, these are the dimensions. The DNA strand shows the polarity, that is poles of the DNA strand are very important. The DNA strand, one strand has at one end 5' end, on the same strand at other end there is 3' end, while other strand which is descending has 5' end and at the other side it has 3' end. Why we can say the polarity or 3' 5' end? In DNA, 3' end and 5' end are so close on each side because of the structure. See, why we can say 3' 5' end? Depending upon this structure, we can clear that. Here, consider this is the strand which is ascending strand. Suppose now this strand has this carbon number 3rd where OH group is free. Okay, so that the at carbon 3 OH group is free. So this strands, this end is called as 3' or carbon 3 end. Whereas same strand when we go upward, we see that this is the phosphate group 3 at 5th carbon. So that to the end of this same strand is 3' here or carbon 3 here, 5' or 5th carbon 5 end. This is the polarity of one strand. So that opposite strand is also having here 3' end because OH group is free at this end, and this is the 5' end because phosphate group is free at the 5th carbon. So both the strands have 5' and 3'. Here also 5' and 3' end, but they are opposite in direction. So on a DNA molecule closely we see that 5' and 3' end on either side or each end to the. Okay. Now this is the polarity of the DNA strand. Now this Watson and Crick model explain one more characteristic feature, that is major and minor grooves. Now see, grooves, this is the major groove and this is the minor groove. Groove means a depression. Due to twisting of the DNA strand, they alternately a deep depression called as major groove, altered by a shallow depression that is called as minor groove. Grooves are also formed here in DNA. Now, in this way, we studied here in this period the 3 points of the topic, that is first the DNA as the genetic material, the second modern concept of the gene, and third the eukaryotic DNA structure or Watson and Crick model of DNA. Okay.